Hello everyone, this is Mr. Fermanik. We're going to do a little notes today. With these notes, we're going to go a little bit quicker than normal because this is a video and you can always go back if you need to, or pause the video if you need to catch up on your notes. We're going to mainly focus on today is protein synthesis, specifically something called transcription. Before we get into protein synthesis, I want to do a little review on DNA. Remember, DNA is double-stranded. It has two strands that form a helical shape, so kind of like a spiral staircase. It has the nucleotides A adenine, T thymine, G guanine, and C cytosine. And the backbone of DNA contains a phosphate and a deoxyribose sugar. So if we look at the images that I have down here, I mainly want to focus on this one on the left that you have in your notes. Is remember the middle is where your base pairs are, and when they bind together complementary, it's formed what's called a complementary base pair, like A to T, G to C. In between those are hydrogen bonds. Remember, hydrogen bonds are very weak and easily to break, and that's going to come back to play a little bit later. Now, on the backbone, between the sugar and the phosphates, are covalent bonds. Covalent bonds, remember, are very strong. They are strong because they hold the order of the nucleotides or the order of nitrogen bases the same, because that's how DNA stores genetic information. DNA, remember, contains all the genetic information for an organism. It has all of the recipes to make you. Think of DNA as this big, huge cookbook. It has all of the recipes that make up you. Hair color, hair texture, eye color, skin color, traits all about you. And the DNA never leaves the nucleus. It's located there and it never leaves the nucleus. With this, think of this, if you had this master cookbook that had all of the recipes that are known to man, it'd be a pretty big cookbook, and it's probably going to have to stay in your basement because it's going to be so big that it can't even stay on the first floor. So how do we use this information? How do we use this DNA to make us? Well, DNA has segments on it that code for certain traits. And you have a segment of DNA that'll code for the type of hair you have. These segments are called genes. A gene is a segment of DNA that codes for a trait. A gene is kind of like a recipe in this cookbook. Like you have recipes for certain items, like key lime pie, you have a recipe for. Now these genes have these instructions to make proteins. That's what actually is made in this process that eventually becomes our traits, becomes what physically you see is an accumulation of proteins that are made by the DNA. Now, when a gene is used to make proteins, it's said to be expressed. Like if you were using a recipe to make some kind of dessert, you're kind of expressing that recipe. So when a gene is expressed, it's called gene expression. Now, to kind of go back to the previous one, when you use a gene to make something, you make proteins. So you're creating proteins in this process. So another name of gene expression is protein synthesis. To synthesize something is to create. So you're creating proteins. So protein synthesis is the same as gene expression. So let's talk about protein synthesis. It happens in two main steps. The first step is transcription. The second step is translation, which we'll get into tomorrow. Now, these two processes, the first one, transcription, starts out in the nucleus, and translation occurs in the cytoplasm. So protein synthesis starts in the nucleus and finishes in the cytoplasm. So like in this image right here, you have the DNA, you have transcription, this is all occurring in the nucleus, and translations occurring in the cytoplasm. So let's talk about transcription. It's the first step of protein synthesis. The first thing that happens is DNA is unzipped by an enzyme called helicase. Helicase breaks the hydrogen bonds between the complementary base pairs, kind of unzipping the DNA, opening it up. Now, the next thing that happens is there's nucleotides that are floating around. So like adenine, nitrogen base that's connected to a ribose sugar and a phosphate, are just floating around, and they'll be matched up complementary to the DNA by another enzyme called RNA polymerase. Now, what it starts to make is this single chain called messenger RNA, or mRNA. So if we look at this, you have the DNA here. You have helicase that's already broken the hydrogen bonds. Now you have this other enzyme, RNA polymerase, that's matching up nucleotides to start forming this thing called an mRNA. So let's talk about mRNA. It's a type of RNA, we'll learn about a couple more, that encodes the information for the synthesis of proteins and carries it to the ribosomes from the nucleus. That's a lot of information right there. Let's think of it this way. DNA is this master cookbook that you have in your basement if you wanted to make something. So let's go back to my key lime pie analogy. You're not going to go downstairs, rip out the recipe, and then go upstairs and try and cook with it, because if something happens to that recipe, you lose that recipe forever. Well, mRNA is kind of like if you went down into the basement and you made a copy of that recipe. It's a copy of the genetic information that's on DNA, whereas if you went down and made a copy of the recipe, it's a copy of the information in that recipe. Now, a couple differences between mRNA and DNA is mRNA is single-stranded and has a ribose as the sugar, not a deoxyribose like DNA. It contains the nucleotides A, G, C, and U, uracil, and uracil replaces thymine, so there is no thymines in mRNA, just uracil. And you get complementary binding between the DNA and the RNA. So if you have DNA going to RNA and you had the DNAs of A, T, G, and C, if you look here, A would match up complementary to U, T would match up complementary to A, and G would match up complementary to C, and C would match up complementary to G. So if we look at this, let's say we have a sequence of DNA here, and we're doing transcription to make mRNA. You can see A is complementary to U, C to G, G to C, A to U, T to A, and A to U. And if you look at the next sequence, CCC, remember C's match up G, so complementary would be GGG. Now if you look at the next sequence, we have TGA, remember T goes to A, G goes to C, A goes to U, you get complementary base pairing that matches up. Now let's take a look, if you may want to stop it and do a little practice right here. Okay, continuing on. The product of transcription is a workable copy of the information in DNA. Kind of like if I said you went down to transcribe, that means to make a copy of your recipe in the basement, you would have a workable copy of that recipe. Now, this workable copy is able to move from the nucleus to the cytoplasm. Like if you went down and made a copy of that recipe, you can now take that workable copy of the recipe up into the kitchen and actually make whatever it is you're going to make. So this is going to end us for transcription. Tomorrow we'll learn about the second half of now we've got this mRNA in the cytoplasm. What happens to it next? All right, so now would be a time to watch the other video about what you're doing for today. Have a great day.